Hi there, I'm Susan Lindquist, and I'm a member of the Howard Hughes Medical Institute, and I work at MIT and at the Whitehead Institute. I'm here to tell you about a variety of different problems in protein folding. In this particular lecture, I'm going to talk to you about protein folding and how it manifests in a wide variety of human diseases. So let me first set the problem for you. Proteins come out of the ribosome as long linear strings of amino acids, and they have to fold up into these incredibly complicated shapes in order to do their jobs. And they do just about everything inside of living systems. And the problem, the difficulty in getting those folds exactly right, and they have to get them exactly right, or the protein either won't function, which is bad, or it'll go off and do renegade bad things interacting with the wrong kinds of proteins, and that's even worse. The problem with them reaching their final functional states with these complicated folds is that they have to do it in the insanely crowded environment of a living cell. So this wonderful movie by Adrian Elcock gives you an example of how crowded the cell is and how jumbled the proteins are and how they're banging into each other all the time. And this crowding and chaos and energy really is part of life, what makes life possible, but it also means that all living systems are kind of on a knife edge of vulnerability because it's incredibly kinetic in there. In fact, that movie, in order to be really accurate, would have to be sped up by 3 million-fold. So you can imagine that if there are any perturbations in the system, proteins will wind up starting to bump into each other in inappropriate ways and start sticking to each other and making a mess. And that happens with all sorts of different stresses. It can influence the process of evolution, and it very strongly influences every aspect of human disease. So you have a very visceral feeling for what protein aggregation is in your own lives. This is it. These are proteins, and they look perfectly fine. This is what happens when you apply heat to the system. The proteins aggregate. And of course, that doesn't happen in your body, but just a little bit of that is actually happening all the time. And if it doesn't get corrected, it can spell absolute disaster for living systems. And so cells use a tremendous amount of energy and lots of different proteins to actually try to keep the state of their proteins like this, clear, beautiful fluid, and to prevent that kind of protein aggregation. And I first got into this problem when I was working on something called the heat shock response. The heat shock response is universal, just as the protein folding problem is universal. And it works like this. You can see in yeast on the far side there, we've taken 2 aliquots of a culture of yeast that are growing perfectly happily at normal temperatures, and we exposed them both to high temperatures. But for one of them, we first gave the cells a mild pretreatment at 39 degrees, so just half an hour. And you can see the difference that the pretreatment made in their ability to survive. The middle there is a similar experiment done with Arabidopsis seedlings. And this experiment here is one done with tumor cells. And it's been done with every organism you can imagine. It's universal. Pre-treatments allow protection from much more severe stresses. So what are the cells doing? What's happening in those biological systems to get them prepared for these difficult, stressful conditions? They're making heat shock proteins. So this is a labeling of the proteins that are made by cells at either 25 degrees or 39 degrees. And the different bands on that gel are just spread out according to the molecular weight of the proteins. And you can see that there are massively new proteins that are made in just tremendously large amounts. They just stick right out at you on the gel. They're called HSPs, for heat shock proteins, because they were first found in response to heat. But we now know that actually they exist and they are induced and made at higher levels in response to just about every stress you can think of. So higher temperatures, osmotic stress, changes in pH, changes in energy balance in the cell. They fix those problems with protein folding. They either prevent the proteins from misfolding in the first place, or they take proteins that have been damaged and have aggregated and start to get into a coddled egg sort of state and get them right back out of it. And every organism on the planet uses this same response. This gel that I'm showing you here happens to be a gel of yeast proteins and the yeast heat shock response. Bacteria, plants, mammals, we all do it. Birds do it, bees do it, even educated fleas do it. So it's very universal, and it's very broadly used survival response. It's used not only in response, as I mentioned, to heat. It protects organisms not just from heat. It protects organisms from all those other stresses I told you about. And this manifests many different ways in biology and in, as I mentioned, every different aspect of human disease that you can imagine. And we actually work, because we started out working on protein folding a long time ago, and it had this ramification in different ways, we actually found ourselves working in all these different areas of biology. So I'm only going to tell you one little bit about how this protein folding problem interfaces with infectious diseases. But I'll tell you a little bit more about what we're doing with cancer biology and neurodegenerative diseases. So this is an example of an organism, a pathological organism invading us through the bite of a mosquito. This is a malaria parasite. And you can see that when the mosquito first bites it, of course, the organism undergoes a big change in temperature when it comes inside of us. And so that causes protein folding stress. It mounts a heat shock response to help protect it. And then in its life cycle, this organism is moving around and going to different parts of the body. And as it does that, our bodies recognize that they're having problems with protein folding because of the heat stress. And our bodies try to make it even worse for the organism by mounting massive fevers. And so this is a constant feature of all pathological organisms that invade us and change from one temperature to another. We actually are only working a little bit on malaria, but we have a major project going on where we're trying to block the ability of fungal organisms, fungal pathogens, to use this heat shock response to protect themselves when they get inside of us. Unfortunately, I won't have time to talk to you about that today, but I do want to talk to you about aspects of protein folding in cancer and neurodegenerative diseases. So with respect to cancer and neurodegenerative diseases, we are actually between a rock and a hard place, because it turns out that cancer cells and, in fact, those infectious organisms, as I just mentioned, are using their survival response to help kill us, empower them to get through all kinds of stresses and power on through those stresses and kill us. And oddly enough, even though our brains during these neurodegenerative diseases are also experiencing misfolded proteins, the massive aggregates of proteins I'll show you in a minute, in fact, that occur in our brain with these diseases, those usually should signal, uh-oh, our proteins are in trouble, let's mount a heat shock response. And for some reason we don't quite understand, they don't do that, or that they at least don't do it very well. So we're kind of in a mess right there. In that balance of cancer and neurodegeneration, one of the major players, and there are more and more of them that are coming out now, but one of the major players that we've found that regulates that balance between health and disease, two different diseases at these different ends, health being in the middle, is something called the heat shock factor. It's the master regulator, that heat shock response I just showed you. It's the protein that goes into the nucleus, binds to those genes, turns them all on, and gets the response going. And so it's a pretty powerful mediator of this balance between health and disease. So our first understanding of how this heat shock response, this classic heat shock survival response, plays a role in cancer was when we were able to obtain a mouse that actually had a mutation in that heat shock factor. It couldn't mount a heat shock response. And that mouse was actually more susceptible to heat stress and other stresses like that. But it was perfectly fine in terms of living a normal lifetime. And as long as it was in a relatively controlled environment, it was just fine. And Eva Benjamin, who'd made this mouse, was kind enough to share it with us. So we looked at how that being unable to mount a heat shock response like that might affect the susceptibility to cancer. And it had a pretty big effect. So this is the first major experiment that we did. What we did was we shaved the backs of these mice and we painted them with a mutagen and a tumor-promoting agent. And you can see that the normal mouse is responding to this very classic cancer-causing agent by producing skin tumors. And these skin tumors, as you're seeing them at a midway stage of the disease, but they got worse and worse and worse, and they eventually killed the mouse. The mice down here are genetically identical to these mice up here. The only difference is that one heat shock transcription factor has been mutated in the genome, and it can't mount the heat shock response. And you can see that that profoundly protects the mouse from cancer, because the cancer cells are actually using that survival response to allow them to grow in an uncontrolled way and to invade other systems and other places in the body where they normally don't belong and where they have encountered stressful environments. So that's how it played out in the mouse, and it turned out that we studied several different causes of cancer. This particular cancer has a particular pathway is perturbed that's kind of a classic pathway in cancer biology. But there are other equally important pathways in cancer biology, and we looked at several of them. And in each case we looked at, there was a big difference in the incidence of cancer. Not being able to mount the heat shock response meant those tumors didn't develop as much and they were not as invasive. In fact, there was this difference in the number of tumors translated into an incredible difference in lifespan. These mice up here died, these mice did not. And that happened again and again with these different cancer models, and it's been shown by other laboratories now as well. So that's mice. What about humans? Well, one of the things we did with human to try to see whether this was really relevant to human beings was to take a variety of different cancer cell lines from humans, and lines that could be grown in culture and manipulated easily, and we knocked out their ability to make a heat shock response, and in fact, they started to die. If we did that with a normal cell and kept it under fairly normal conditions, they were fine. But when we did that with cells that were cancerous from all sorts of different cancers, from all different sorts of causes, they died. But you've got a problem here now. We're looking at mice and we're looking at tissue culture cells in a dish in a very abnormal environment. Can we get any idea, can we get any evidence that this really matters in a human being? And so what we did, one of the pathologists working in my group and another pathologist working in Bob Weinberg's group, Tan Inche and Sandro Sanagata in my group, looked at whether or not when you took out tumor samples directly from a patient and stained them with an antibody that would react with the heat shock transcription factor that I just told you about, would there be a manifestation of the activation of that transcription factor in the human tumors taken directly and immediately from the body. So here you have a slice of human tissue taken from a breast cancer victim. And you can see the brown staining is HSF. The blue is just a generalized protein stain. And you can say we've caught the border between the normal cells here and the tumor. And that's a very special place to be, because we don't have to worry about the difference in staining being due to a difference in the fixation of the tissue or whether the antibody was able to penetrate the tissue. We know that the tissue, the normal tissue and the tumor tissue, which are right next to each other, are being treated in exactly the same way. And so here we see that HSF, the brown staining, is in the cytoplasm. And that's because that's a normal cell, they're perfectly happy, and they don't need their heat shock response. But in the tumor, you can see that the sort of deranged-looking cells have brown, much higher level of that brown stain, much higher level of the heat shock factor, and it's moved from the cytoplasm into the nucleus, where it does its business in transcribing a whole variety of survival genes. So that's breast cancer, that's pancreatic cancer, that's colon cancer, and that's lung cancer. In fact, we've stained dozens and dozens of different types of cancers, and we've seen it in every single one of them that we've looked at. And moreover, what's interesting about it, and a little bit unusual when it comes to looking at cancer markers, is that along the tumor, throughout the tumor, the staining is quite uniform and even, as though the whole tumor is really using that response. So that looks interesting. This response is being turned on in the cancer cells and not in the normal cells that are right next to them. But does it really make a difference? Well, of course, our experiments with mice and our experiments with tissue culture cells would suggest that it's making a difference. But can we get any evidence that it really is? we can actually. We can because of a wonderful group of studies that have been done over the course of many, many years by dedicated clinicians. And the first study that we looked at was something called the Nurses' Health Study. Nurses are a particularly wonderful group of patients because they're very responsible. When they enroll in a study, they will come back year after year after year for their examinations, and so that you can track them and find out what happens to them. So this is a particularly valuable study, and it's the nurses that enrolled in this study have been examined for many different pathological conditions, and that study has told us a great deal. In this particular case, what we did was to get a hold of samples from the nurses that were first enrolled in this study 25 years ago. And we stained their initial samples on their first biopsy. We stained them for whether or not they had a high level of HSF or a low level of HSF, and whether it was in the nucleus or whether it wasn't. And you can see that, and then the pathologists stained these slides blind. They had no idea what had happened to those nurses, and they scored them for high, medium, or low levels. And then we decoded the data because the survival records had been kept for those women 25 years later. And sure enough, if in the women who had a low level of the heat shock response active in their tumors, they were much less likely to die than the women that had a high level of HSF in their tumors. So this is kind of an amazing difference in these populations when you realize that these women were treated in different ways all over the country, in different clinical settings and clinical centers. So to see such a large difference in their survival really would suggest strongly that the heat shock response, when it had been on in their tumor to begin with, it helped those cancer cells to survive and helped to drive the malignant state in a very unfortunate way for them. Now, was this just a one-off from breast cancer? No, we were able to actually do something similar in lung and colon cancer. And these are the three major causes of cancer in the developed world, the three major killers. And we think, of course, it's probably not just these diseases. And not just these cancers, we think it's actually much, much broader. But certainly in these, we're able at least to get a correlation between the activity of this heat shock response and the outcome in terms of malignancy. And that allows us to ask, well, can we do something about it? And that's what we're trying to do. We're wondering if we can use it diagnostically. In other words, if in fact when someone is first diagnosed as having cancer in the clinic, might staining for the heat shock response help clinicians to decide whether or not that patient should be treated aggressively or not aggressively? It's often a very, very difficult decision to make. And we think that you might not want to treat patients with an aggressive chemotherapy agent that might wind up inducing a heat shock response, inducing the survival response, if the tumor in the first place doesn't have it. Don't know if that's going to work yet or not, but that's the logic of what we're trying to do. And then, okay, that's diagnostics, maybe. We think it's promising at least. But then could we ever learn to control it in human cancers? Could we surgically block with chemical agents or with antibodies, we surgically block the activation of that response in tumors where it's already active and try to turn it off? So that's another thing that we're trying to do. So how can we use that combined knowledge therapeutically? Would be manifested in lots of different ways, but one thing you can imagine is you find out whether or not the patients have that heat shock response. And you really only treat the patients that do have the heat shock response on with that particular agent, because you don't want to treat people unnecessarily. So those are the ways in which we hope to use that response and use our knowledge of that response. It's still very early days, but that's how basic science really tries to interface with difficult clinical problems. So you might think that this powerful survival response that the cancer cells are taking advantage of, that our nerve cells are not taking advantage of, maybe if we turned on that survival response, it might be a great therapeutic strategy for all of those different protein folding diseases. It could maybe work on all of them. I don't think so, however, and the reason is because I think turning up that response when it's not normally needed might make the brain more susceptible to development of cancer. So in terms of treating neurodegenerative diseases and trying to find some solution to these horrible diseases, we're going about that protein folding problem a little differently. We're actually trying to attack the individual pathologies that are caused by individual proteins that misfold in those diseases, so we can have a much more targeted sort of surgical strike against those diseases. And I'll be talking about that in my next lecture. Meanwhile, I just want to thank you for listening.